Good morning, everybody. My name is Nathaniel Penrod, and I'm with Michigan State University. I'm a graduate student in the Coastal Processes and Geomorphology Lab in the Department of Geography, Environment, and Spatial Sciences. And I'm here presenting a project that we've done in collaboration with the National Park Service and Northwestern College in Wisconsin. I'm going to be shifting gears a little bit from the presentations we've had previously at this conference, focusing on applications of high resolution bathymetry and some cool stuff you can do with it once it's collected. Specifically, I'm going to talk about a project we've been working on to model the risk of Dreissenid mussel colonization in and around unionid mussel habitats in Lake Superior U.S. national parks, primarily focusing on the Apostle Islands, but incorporating a little bit of information from Isle Royale National Park as well. So today we'll start off with a little bit of background on the project, talking about why invasive mussels are a problem in our parks, previous monitoring efforts that the National Park Service has done, and then how we've tried to basically systematize our monitoring efforts so that they're more consistent and repeatable. We'll overview some suitability modeling approaches and two different models that we've used to predict where mussels are more likely to colonize, share some initial results, and then as a conclusion, go over some lessons learned and what our next steps are. So as I'm sure you are all aware, the Great Lakes have an invasive mussel problem. Beginning in the 1980s, two species of Dreissenid mussels were introduced to the Great Lakes, zebra mussels and quagga mussels. I'm going to be talking more about zebra mussels today because that's the one we've had more of a problem with at the Apostle Islands. But they're both nuisance species and they have pretty profound negative ecological impacts on the Great Lakes. The two that the National Park Service was most interested in were competition with native unionid mussels and also the fouling of lake trout and other fish species spawning habitat grounds. So far, these invasive mussels have spread less quickly in Lake Superior than they did in the lower four Great Lakes, primarily because of colder mean lake temperatures in Lake Superior and also because of less availability of nutrients like calcium and phosphorus, which are essential for healthy shell formation. So once invasive mussels were first discovered in the Great Lakes, people at the Apostle Islands jumped on trying to get some kind of monitoring program in place, at least have a baseline reference point for what unionid mussel populations were before native or invasive mussels reached the park. So in 1991, there's the first survey of unionid mussel populations at the Apostle Islands. You can see the map of the site locations where they surveyed back then on the right there. But after that baseline survey, they found no zebra mussels in the park and efforts kind of fizzled. So we had a very sporadic and frequent monitoring from 1991 up until 2015 when some park divers discovered zebra mussels on a shipwreck within the park. After that, in 2017, they followed up with another comprehensive unionid mussel survey, and they found that unionid mussel density had decreased at some sites, and they also found zebra mussels in way more places than they thought they would, particularly around docks, which isn't much of a surprise, but also in a wide variety of substrate types that wasn't previously expected. So after that 2017 survey, the Park Service began to develop they recognized the need to develop a more systematic monitoring approach to how they were going to go about monitoring for the presence of invasive mussels and also communicate where those mussels are and what the public can help do to avoid them spreading more and more throughout the park. So one of the things they thought would be really helpful in terms of systematizing monitoring efforts would be to incorporate bathymetry. So it's no surprise to anybody in this room that bathymetry is really a cornerstone of good benthic management. Without good bathymetry, we don't really know what's out there, and it makes it really hard to figure out where we're likely to see mussels now and in the future. So lucky for us, the National Park Service in the Great Lakes had been doing high-density multi-beam surveys from 2010 through 2018. And these four parks shown here on the slide, like I said, I'm going to focus mainly on the work at the Apostle Islands. But combined with some topo bathymetric LiDAR to kind of fill in the nearshore gaps that we got from NOAA's data access viewer, we were basically able at the Apostle Islands to create a complete bathymetry mosaic for park waters and then some areas outside of park managed waters as well. One reason why bathymetry is really important to predicting the presence or the potential for mussels to colonize is that both zebra and quagga mussels have specific depths where they prefer to colonize and where they're more abundant. So zebra mussels tend to colonize in shallow waters, typically 30 feet or less is where you're going to find the densest zebra mussel populations. Quagga mussels, on the other hand, are a bit more of a generalist species. They've been found deeper than 100 feet in Lake Ontario. So it's a bit harder to model where quagga mussels might colonize because of their broader depth tolerance. So we focused more on monitoring zebra mussels since those were the mussels that had been found in the Apostle Islands, and they also have a much narrower window of environmental conditions where they can exist. The other piece of data we wanted to take into account to help us develop a more systematic monitoring approach was substrate composition. Unfortunately, we had two big challenges with this. One, we had very patchy and incomplete backscatter due to a number of issues with the early multi-beam surveys, which really hindered our ability to differentiate between hard and soft substrates in our multi-beam surveys. And we also had little available ground truth information. There were some areas of the park where we had done underwater video missions with a sea viewer camera, but it definitely wasn't enough coverage to be able to build and validate a substrate map. So with those challenges, we kind of set out with two objectives that we wanted to achieve. The first was we wanted to model substrates most suitable for initial zebra mussel colonization. And then the second one was we wanted to target the observation of adverse ecological impacts of invasive mussels on native unionic mussels as well as lake trout spawning grounds. No problem. Okay, great. So for our study sites, like I said, I'm going to focus more on the Apostle Islands, but we've also begun a pilot program at Aotearoa as well. So at the Apostle Islands, the park staff identified 20 potential sites where they thought it would be good to monitor for the presence of invasive mussels based off of previous diving efforts as well as local expert knowledge. And we did a similar thing at Isle National Park and picked 13 areas where we thought it would be good to set up monitoring locations. So once we identified those, we started building models in GIS to help us narrow down within those study areas where suitable substrate for zebra mussel colonization might exist. So in 2021, this was kind of the first iteration of a model that we built. So we took bathymetry, terrain ruggedness, and an unsupervised classification of a principal component analysis of bathymetry and other derivatives from bathymetry. And we basically just did a threshold analysis, a binary threshold analysis, where if you were within, if a cell was within a set of conditions, it was identified as suitable. If it didn't meet all three of those conditions there for depth, ruggedness, and the unsupervised classification, it was classified as not suitable substrate. From there, within all of the sites identified by the suitability model as potentially good substrate, we generated a bunch of random points throughout those areas, and then we selected our field sites from those random areas, or from those random points. So this was a good first attempt, but after we finished this field season in 2021, we got some feedback from the people that were doing the surveys, and we thought, there's got to be some more literature out there for different conditions other than just depth and kind of a rough approximation of terrain ruggedness that would help us improve and kind of hone in these models a bit more. And it turns out after we did a literature review, there are quite a few other variables that impact the suitability of a site for zebra mussel colonization. So in 2022, we took another stab at a suitability model, incorporating two new key data sets. The first was a benthic geomorphology classification that we generated using the bathymetry and reflectivity-based estimator for seafloor segments. I know that's a mouthful, but luckily we have acronyms, so BRESS. This was a tool developed by researchers at the University of New Hampshire, and previous presentations at this conference in past years have talked about efforts that the Park Service has done to incorporate or figure out how to use high resolution bathymetry to create these kinds of geomorphology classifications using breasts. And really the advantage of using breasts for this application is that it's almost completely automated. So there's very little ground truthing data. Actually, you don't need any ground truthing data to use it. And it allows us to really hone in on geomorphological features relevant to species habitat suitability. So for instance, it does a really nice job of pulling out ridges where fish spawning sites are more likely to occur, and also flatter areas that are more likely to have the mud and the silts that native mussels can burrow into. The second data set that we incorporated into this model that was really important was an average weighted fetch model. So fetch, as many of you probably know, is the distance that wind blows over open water before encountering some kind of obstacle that would divert or slow down the flow of the wind. And fetch, from the literature, we found it's a really influential factor for the suitability of habitat for native unionid mussels and also for lake trout spawning grounds. So we created an average weighted fetch model. So basically what that means is we looked at 30 years of wind data from the National Data Buoy Center, and we did a frequency analysis to say, okay, from each of the cardinal directions, how often is the wind blowing from the north, the south, the east, and the west, as well as the subordinal directions. And then we were able to weight the average fetch of any particular grid cell using the frequency that the wind typically would blow in from any given direction. So here's what those data sets look like. So on the left there is an example of the geomorphology classification from Sisquit Bay at Isle Royal. There are 16 total classes of geoforms. And you can see there in blue these ridges that are getting pulled out by the tool really nicely, as well as those grayer flats. Those turned out to be really helpful features for us to capture. And then on the right there is the average weighted fetch model for the Apostle Islands, with the pink cells there indicating overall higher fetch, and then the light blue cells lower fetch values. So this is what the workflow for our second model looked like, a bit more complicated. And really the thing I would like to draw your attention to here is how we were able to integrate suitability models for different species we were interested in into kind of a master monitoring suitability framework. So within ArcGIS Pro, they have a great suitability modeling tool, which allows you to select variable weights and different weighting schemes for all of your input variables. So we basically created a suitability model for native mussels, invasive mussels, and lake trout spawning areas. And then we threw all of those suitability models into the ArcGIS Pro suitability modeler again, all with equal weights, to basically create a monitoring suitability model. What that allowed us to do was to look not just at areas that were potentially suitable habitat for these species, but also look at interactions between species and areas where competition for resources was likely to occur. And then after we generated that final monitoring suitability model, we followed the same process of creating random points in those areas and then identifying potential field sites. And then once we got out into the field, divers from the National Park Service did a bunch of scuba diving transects. So they would navigate to a point previously identified by the models as being suitable for monitoring. And from that point, they'd dive 30 meters north, south, east, and west of the site. And along those lines, within, I think, a 2-meter buffer of that transect line, they would record the substrate type and then also the presence of any invasive mussels along those transects. So our initial results from this project, using the first model, we basically found that the first model, because it was just a simple binary yes, no, is it suitable, is it not, we had really constrained potential field sites. And as you can see here, one of the variables that really constrained the analysis was that terrain ruggedness component, in terms of identifying places where zebra mussels were likely to colonize. The reason why terrain ruggedness was so constraining is we really tried to look at only the most rugged surfaces The assumption there being that rugged surfaces are more likely to be hard, cobble, boulder, bedrock, which is where zebra mussels tend to colonize first in an area. The other limiting factor was we only looked at depths between 5 and 15 meters to be safe for scuba diving operations. And we also left out the consideration of any kinds of other substrate types besides hard cobbles, boulder, and bedrock that could potentially be suitable areas for zebra mussels to colonize. However, the advantages of this approach were it was very simple and time efficient. And like I mentioned, we had some very specific field work targets come out of this. So with the second model approach that we just finished implementing this year and that helped guide our field campaign this past summer, we got a more holistic picture, not just of inside of our individual study areas, but park-wide, what does suitability for these different species look like? So this was our invasive mussel potential suitability map for the Apostle Islands. You can see there's several hotspots being identified, kind of on the western side of different islands. This was our native mussel potential suitability map. You can see here down south around Long Island, there's definitely a hotspot down there, as well as south of York and Sand Islands, and west of Outer Island up there in the upper right-hand corner. And then for lake trout spawning grounds, there's definitely seems to be a collection of highly suitable sites near Gold Island Shoal, also around the York Sand Island area, and then in the shoals off north of Outer Island at the top of the map there. So we threw all three of these models together into a monitoring site model, which looked like this. And basically this showed us the sites where there was likely to be suitable habitat, both for invasive mussel colonization, but also for lake trout spawning grounds and for native mussels, so that we could try to monitor for the competition between those species. Some limitations of this approach that we found were because all three of the initial intermediate models use the same variables, the models aren't independent of each other, which creates a little bit of a spatial autocorrelation issue. And the evaluation, or the selection rather, of the weighting scheme for each of these models is somewhat subjective and was mainly based on the input from field workers and also from park staff. Some advantages of this model lean approach that I've already alluded to were it incorporated more variables from the empirical literature known to impact habitat suitability, and it also helped focus our monitoring on places we're more likely to see interspecies communication or competition for resources. And over the entire park, it kind of helped highlight hot spots and cold spots, which was really helpful for park staff to be aware of. So from our scuba diving sites over the past 2 field seasons, at Isle Royal, only 4 sites were eligible to be dived this past year, mainly because we're still recovering from the impacts of COVID and our dive team was spread really thin. But the great news for all 4 of those sites were that there were no zebra mussels. So far, as we know, Isle Royal doesn't have zebra mussels at the sites that the model predicted it might. So at the Apostle Islands, it's a little bit of a different story, so I'm going to focus the rest of our time addressing our results from there. So we had 36 total sites where we did scuba dives. All of those sites were originally selected using the first modeling approach, and then we confirmed their potential suitability using the second model. Of those 36 sites, 86% of them had suitable substrate for invasive mussel colonization. You can see a picture of what park divers identified as suitable substrate up there at the top on the right-hand side. Only 11% of those sites had substrate that was deemed not suitable for invasive mussel colonization, which you can see in the middle, and then 3% of our sites were potentially suitable, where there's a mix of hard and soft substrate types. And then across the Apostle Islands, 39% of those dive sites had zebra mussels and 61% did not. And you can kind of see a pattern of up to the north of the Apostles, and then also a little cluster kind of in the middle around Oak Island Shoal, where there were zebra mussels present. So to wrap things up here, some of the key benefits that we've gotten from taking the suitability modeling approach were, without having ground truth data and without having high resolution substrate maps, we were able, at least at the Apostles, with an 80% accuracy rate to identify a suitable substrate for invasive mussel colonization, which, without having to put in the time or the effort to do a really intensive field campaign to collect that ground truthing validation, with basically just me sitting behind my desk using ArcGIS, I think it was a pretty phenomenal result. We also doubled the efficiency of our diving operations, primarily by reducing the amount of time it took for our dive teams to find places they think they might want to dive. And because what was common in the past, especially in the 2017 survey, was the team would go out, they'd set up at a site, and they'd look down and kind of be like, okay, I think this is a good place to dive. They'd get down to the lake bed and it would be completely sandy. So that's a wasted opportunity to, and it really limited the spatial coverage of those surveys. So having a more targeted approach beforehand, before you go out into the field, meant that we could get a much higher spatial coverage of the parks, having done this suitability modeling beforehand. So lessons learned in moving forward, as could be expected, we learned that this model building is a nonlinear and highly iterative process. We had a lot of iterations, particularly of the second model, where we facilitated impact from input from different park staff and people who were involved in field operations to kind of help us fine-tune how we were parameterizing our weighting schemes, which was very helpful. One area where our models did underperform in terms of predicting suitable substrate for zebra mussels was in embayments, where the typical substrate was a hard-packed clay overlain by a thin layer of silt or mud. If you look at the bathymetry data, just visually, it looks like it should be a somewhat harder substrate, but that was not the case. So one of the things we're going to do moving forward is try to figure out a way to better tune our models to not pick up on those kinds of embayments as suitable zebra mussel substrate. The other big lesson we learned is that risk and suitability are not one and the same. So in communicating these results so far to project partners, both at the Park Service and as we start communicating these results to the public, we learned we have to be very careful about saying that these models show suitability or potential suitability and are not necessarily showing the areas at highest risk, because risk has way more factors that go into it than just saying we think these are the areas that are most potentially suitable for these kinds of species to grow. And finally, some next steps that we have are we're going to analyze the patterns of the zebra mussel distributions, specifically at the Apostles, trying to understand what are variables that are causing them to kind of be on that north edge of the park, if there's any kind of current or flow conditions that might be contributing to those groupings that we're seeing out there. And then we're also starting to work with park staff to use the native mussel model and the lake trout spawning ground suitability models to advance separate monitoring efforts that are going on for both of those species right now at Isle Royale and also at the Apostle Islands. So finally, we'd like to acknowledge the Great Lakes Restoration Initiative for funding a lot of this research. Also from the Park Service, Laura Bender, who is our hydrographic surveyor for this project, as well as park staff from Isle Royale and the Apostle Islands. And then from Michigan State University, kind of coming in and helping with analysis at the last minute for these models, Drs. Ethan Tierkoff and Aaron Bunting. So with that, thank you for listening, and I'm happy to take any questions.
It's a general interest question. I mean, how big is task is that understanding these assessments either happening, vast areas yet to assess, or cadence of how to track this over time to see sort of progression of the data?
Yeah, so are you asking about the scale of the monitoring efforts going forward, essentially? Yeah, so the plan right now is to try and build up to being able to do think within each season, we can really only do about 20 sites if nothing goes wrong with weather in terms of doing scuba diving. So one of the big things we'd be looking at in the future would be other kinds of field work. So drop camera or some kind of AUV, ROV system that could help automate some of that field work. Because if you have to send divers out, there's really only a limited number of sites you can get to at these parks each year. And the goal would be as we stack field seasons to go to different areas of the park. So for instance, this past summer, divers may have targeted areas kind of inside the middle of the Apostle Islands, and then next field season, we tried to go maybe to the eastern side of the park instead of going back to sites we had already done until we can get a more full spatial coverage throughout the entire park, which is really difficult because both the Apostles and Isle Royal are massive. So it takes a lot of work to get divers out there to really do the in-depth field assessments that we're looking to do.
I'm wondering if you there any lessons learned from the bathymetry aspect that you used for the classification and probably classification. Any feedback for how we can collect that data better to be more suitable for that?
Yeah, so I think the primary lesson we learned was, especially with the early multi-beam data that the National Park Service collected, we had a lot of issues with holidays in the data, trackline artifacts, and also with our sound velocity profiles. We'd have, for instance, even within like a single day of surveying, we'd have two backscatter results that would come back. One would be, really high values of backscatter. The other one is really low, side by side. So because of issues with one, we were still trying to figure out how to use the systems well back in 2010 and 2011. That was one thing. But after those hiccups got worked out, the main thing that was really important was basically the higher resolution that we had, the better. So the highest resolution we could achieve park-wide was a 2-meter resolution. In areas where we had a 50-centimeter resolution, that actually really helps us hone in on, especially incorporating the whole terrain ruggedness component. Having that higher resolution really helps us hone in even on like individual boulders or cobbles, and maybe even different aspects of that substrate that might be more suitable to colonization based on currents and things like that. So that's one of the things we're going to look at in the future. It's how having higher resolution bathymetry data would allow us to maybe incorporate more variables into the analysis that at a 2-meter resolution we still can't.